So then we start with the actual meiosis cycle. And just like with mitosis, the meiosis cycle is the same thing. You want to memorize PMAT, P-M-A-T. And so the first stage that we're working on right now is the P for prophase. So we're in Meiosis 1 right now. So we're going to be in Prophase 1 stage of the Meiosis 1. So we're going to start with that germ cell that I had drawn on the previous page. And it's going to, we call this germ cell 2N. However, as I just went over with you, during interphase, before we get into prophase, those chromosomes have duplicated themselves. So since it was a 2N cell, it's actually 4N just for a short period of time. And so just depending on your teacher, I don't know whether or not your teacher wants you to include that detail when you're answering questions on your homework and on the test. But just so you are understanding what is going on here, I just wanted to point that out. And then we end with this cell that's ready to go on into Metaphase One, which is a lot like the end cell of prophase in mitosis. The centrosomes, the little Mickey Mouse ears, have moved to opposite ends of the cell. They have grown the microtubules. They've started to grow the microtubules out of the little fingers coming out of them, which forms a spindle. A spindle meaning like a cage. So if you have, I'm going to draw this right side up. If you have a centrosome on at the top of the cell and a centrosome at the bottom of the cell. Here, I'll draw the cell wall. And then you have these microtubules coming out into the center of the cell like this. And this is also why I refer to them as fingers, because they end up doing this. And see, it looks like two hands with your fingers interlaced, like you're getting ready to pray. And so this kind of looks like a cage. If you squint really hard, you can see that it looks like kind of like a bird cage. And so this is called, this whole contraption is referred to as a spindle. So you're going to hear teachers in your textbooks refer to this as a spindle, or you could hear them refer to these finger-like projections as microtubules. So just so you know that if you're hearing two different words, we're still talking about the same thing. So more information on Prophase 1. We're still in Prophase 1. I just wanted to write a summary here. The nuclear envelope breaks down. This is just like in mitosis, which is why I wanted you to go back and refamiliarize yourself with mitosis. Remind yourself of mitosis, because this is real similar. So the nuclear envelope breaks down. So see, here's the nuclear envelope in black, the walls of the nucleus. But if you look down at where we end up, there's no nucleus. There's no nuclear envelope or anything. And the chromosomes are just all floating around all throughout the cell. Also, the centrosomes, as we just went over, the centrosomes move to the opposite poles of the cell and start to develop a spindle, there's that word spindle, of microtubules. And finally, and probably the most important thing about this, is that in Prophase 1, in Meiosis 1, Prophase 1, the chromatin, it winds up. Remember, the chromatin is the unwound version of chromosomes, that that messy ball of string in the nucleus. The chromatin, it winds up into the little segments of chromosomes, into two sets of homologous pairs. So that's a total of 4 chromosomes. That's a total of 4 segments each chromosome. So there's going to be 2 pairs of sister chromatids, not just 1 pair of sister chromatids each for each chromosome, like in mitosis. In meiosis 1, in prophase 1, we're going to have 2 sets of sister chromatids, not 1. And what happens since we have 2 sets of sister chromatids, what happens is crossing over can occur. And crossing over is just where a little piece of one of the chromosomes breaks off and changes places with a little piece of the neighboring chromosome, the chromosome right next door. So they just exchange pieces, and so it makes two totally new chromosome combinations. And these are called recombinant chromosomes. So this is really important. So let's go into crossing over a little bit more before we move on out of prophase 1. So crossing over, as I said, it forms recombinant chromosomes. Well, what do I mean by recombinant? Well, recombinant simply means recombined. If you hear recombinant, just think recombined. Recombinant is just a fancy word for recombined, to tell you the truth. And it makes new genetic combinations of chromosomes. So if you look at this picture that I drew, here are the 2 sets of sister chromatids next to each other because they've duplicated in the S phase of interphase already. So there's 2 sets of sister chromatids. And see how they're right next to each other? They're hanging out with each other. And up here, as you can see, one of their arms crossed over, which is why we call this crossing over. They literally cross over each other, and they end up breaking off and swapping out their pieces of their chromosomes. So now this purple these purple sister chromatids have one of them has a little blue part from the other chromosome, and this blue set of sister chromatids, one of them has a little purple part in its chromosome. So now these are not identical anymore. These sister chromosomes, like here's 1 and here's 2 from this pair, the pair isn't an identical pair anymore because one of the chromosomes from this pair has a new little blue piece on the top. And same with this uh, blue pair of sister chromatids. These are not identical chromosomes anymore because one of them has a little purple piece on the top instead. So crossing over recombines the chromosomes, making new combinations. So the chromosomes are all mixed up and have new genetic information in it. So where you could have had a gene coding for blue eyes, but then it crossed over with the neighboring sister chromatid, now instead you have a gene coding for brown eyes. So it makes a different variety. And so that means if a mom and dad have more than one kid, the kids aren't all going to be twins. They're going to look different as long as they're not actual twins. If a mom and dad has a child and then the next year they have a child and then the next year they have a child, these children are all going to look different from each other. And this recombination, it happens a lot. I mean, this is not a rare thing. It happens all the time. And it also, as far as we know in science so far, as of 2022, the crossing over happens at random. As far as we know, there isn't anything going on that intentionally like picks out what is going to cross over and what's not going to cross over. But, science changes. We update our information as we learn new things. So this could be maybe it's not random. Maybe we just haven't discovered the mechanism for it yet. But as far as we know right now, they cross over randomly. So now we're in Metaphase One, the second phase in Meiosis One. Remember, meiosis is just like mitosis. It's P-M-A-T, PMET. It's just that meiosis, since we're dealing with double the amounts of chromosomes, we have to go through the meiosis cycle twice, and mitosis you only go through once, but they're pretty much the same cycles. So metaphase one, we're going to start at the end of prophase one. So here's the cell at the end of prophase one. We have the duplicated chromosomes, so these little double Ks here. So you get, here's one there, and then you get one there. So they're like double Ks back to back. And then we have the centrosomes on opposite ends of the cell, starting to form the microtubules coming down. And there is no nuclear envelope anymore. The nuclear envelope's dissolved, and so all the chromosomes have spilled all throughout the cell and are swimming around all over the cell. So that's where we're starting from at metaphase 1. And then by the end of metaphase 1, all of these double Ks, these double sets of chromosomes, are still in their double form, but they line up still all through the midline of the cell, just like in metaphase of mitosis. It's just that the chromosomes are doubled. So if it was mitosis, you'd have the cell and the chromosomes would be lined up. The set of chromosomes would be lined up like this down the middle of the cell. But we have double the chromosomes. So we have double chromosomes lining up down the middle of the cell. And also at the end of metaphase 1, these microtubules have grown all the way out to the center of the cell and have attached to the one half of the double chromosome. So they've attached themselves to an entire set of chromosomes on their side of the cell. Now, I should also say, and I have it written down here at the bottom, that these chromosomes lined up at the end of metaphase 1 they're lined up randomly. So as you can see, I have purple for the chromosome set from mom and blue for the chromosome set from dad. And as you can see, it's just they're kind of random which goes on what side. So you will end up with some chromosomes from mom going to the left and some chromosomes from dad that are going to get pulled to the left, and some chromosomes from dad are going to get pulled to the right, and some chromosomes from mom are going to be on the right side of the cell. So when we get into anaphase, these chromosomes are going to get pulled to either side of the cell, so we're going to have a lot of diversity in the genetic makeup of the cells made from this. Now, Anaphase 1, we're still in the first cycle of meiosis, so we're in Anaphase 1, and don't forget PMAT, prophase, metaphase, anaphase, telophase. And in Anaphase 1, I just drew the ending result cell on here instead of repeating the drawing from the last slide at the end of metaphase. So we have at the end of metaphase, we have this cell in this format down here. And so this end of Metaphase One is going to be the starting point for Anaphase One. So this cell up here is going to be the ending point for Anaphase One, because during Anaphase One, all these microtubules that have come out and attached themselves to sets of chromosomes have pulled them back home, pulled them back toward the centrosome, and getting them on either side of the cell. So now, if you notice, I even made a note to notice that these are still in pairs, in sister chromatid pairs for the meiosis one in anaphase one. These are still pairs, sister chromatid pairs that are still stuck together, but what we have broken apart are the double sets of chromosomes. So we have one set of sister chromatids going to the right and one set of sister chromatids going to the left. Now, telophase one, including cytokinesis. Here's going to be the end result of telophase one right here. All these sister chromatids have been pulled to either the left side or the right side of the cell. And so now they're not in double chromosomes anymore. They're not in those double Ks back to back anymore. Those have gotten pulled apart from each other. So now we have a set of sister chromatids in the left side and a set of sister chromatids on the right side. And as you can see, the nuclear envelope has formed around each side, each set of the chromosomes on each side of the cell, forming a nucleus, well, forming two nucleuses, one on each side of the cell. So this cell can start this cleavage in the middle, which is the cytokinesis part, and the cell is cleaved in the middle, and you end up with the resulting two separate cells. So here's the nucleus and here's the nucleus. So this separates and divides into a new cell, and this separates and divides into a new cell. So at the beginning of meiosis one, we started out with one cell, and it had 4 sets of chromosomes in it, and now we're down to, we divided that into 2 different cells. Oh, and I should point out, just to remind you again, that these chromosomes or they're still sister chromatids technically. Same thing. It's just you just call them different things depending on what stage of mitosis and meiosis they're in. But these sister chromatids in both of these nucleuses are all there's a good variety among them because they've all lined up. I went over this uh, for Metaphase One. When they all lined up along the middle of the cell, they lined up randomly. Um, so some of mom's were on the left side and some of mom's were on the right side. Some of dad's were on the left side and some of dad's were on the right side. So when they're pulled over to one or the other opposing sides of the cell, you're going to get a variety of both mom and dad chromosomes, sister chromatids from both mom and dad. So that's going to create more of a variety of the genetic makeup in these 2 new cells that were formed at the end of meiosis 1. Now, meiosis 2 We are starting with the two different cells that we made by the end of Meiosis One, by the end of Telophase One. We're starting with, here's cell 1 and here's cell 2. So these have different genetic makeup in it. If this cell went on to contribute to making a child, it would have a different genetic makeup than cell number 1 going on to contributing to making a child. These have different genetic makeup because of the crossing over that I went over about Prophase One and the fact that they just kind of randomly line up. Now, you take both of these cells, and both of these cells at the same time are going to go through meiosis 2. So each of these two cells, after they go through meiosis 2, which is the same thing as mitosis, it's PMAT, and you see, just like in mitosis, these sister chromatids now are just the little X's. There's one chromosome backed up to the sister chromosome there, making little X's. So now we're just dealing with a single set of chromosomes in each of these starting cells. And these cells are going to go through meiosis 2, which is just the same thing as mitosis. We've got prophase, metaphase, anaphase, and telophase, and then the cytokinesis to finally break the cells apart into two different ones. And so this first cell is going to, by the time meiosis 2 is over, is going to have made these two haploid cells, because these little X's, these little sister chromatids, are going to get pulled apart into just one chromosome per cell. So that's just plain old N. There's no 2, just plain old N. These are going to be haploid cells because they're just going to have one set of chromosomes. And this cell number 2 is going to do the exact same thing, and this cell number 2 is going to produce two new cells that are genetically different from each other. So that results in 1, 2, 3, 4, 1, 2, 3, 4 new haploid cells that are all unique, that are all genetically different from each other. And these finally are what we can call the gametes. These are the sex cells, because these are now going to go into their own interphase where they go through the growth phase, and they're going to grow up a little bit, and they are what turns into the sex cells, the gametes, which are either eggs if you're a female or sperm if you are a male. So that's it. I hope that was clear. I tried to be as concise as possible, but I also wanted to make sure that I explained things very well. So this series does have several videos to it, but my goal was by the end of you watching this playlist, you will have a much better understanding of mitosis and meiosis, their similarities and differences. So if you do have any questions, or I didn't go over something about this that you want to see a video on, please leave a message in the comments and I'll get back to you as soon as I can. And don't forget to like and subscribe. Thanks a lot. And good luck on your cell division tests.